Hello, everyone, and welcome. My name is Kristen Dolan, and I'm the Assistant Director of Operations and Strategy for the Benioff Center of Microbiome Medicine at UCSF. Tonight's lecture will be presented by Dr. Sergio Baronzini, a distinguished professor of neurology at UCSF. Dr. Baronzini's research focuses on the genetic and molecular mechanisms underlying complex diseases, in particular multiple sclerosis. The research conducted in his group leverages approaches in human genetics, immunology, molecular biology, bioinformatics, and systems biology. Dr. Baronzini and his colleagues are also the creators of SPOKE, an open knowledge network that aims to integrate all publicly available biomedical information. Dr. Baronzini received his PhD in human molecular genetics from the University of Buenos Aires in Argentina. His talk this evening is titled The Gut-Brain Axis: Gut Microbial Influences on Neurological Diseases. Sergio, the floor is yours.
Thank you, Christine, and thank you for the opportunity to present today. Welcome, everybody. So the title of my talk is The Gut-Brain Axis and How Gut Microbiome Influences Neurological Disease. And given my background and my interest in genetics and immunology, and our focus on multiple sclerosis, which is probably one of the prototypic diseases that intersect the immune system and the central nervous system. I think I'll provide a rather unique perspective on how the gut and the brain might communicate. And this is what is known as the gut-brain axis. And the reason why the gut and the brain are related are multiple, in fact. Here I'm presenting a sketch of what it will be a section of the gut. And here you can see some of the cells that make up the barrier between the blood and the gut. So here's when you will see on the left-hand side, this is where the food will be processed on the gut lumen. And the Peyer's patches are specializations of the membrane of the gut that will allow intimate contact between these cells and the immune cells which are circulating in the blood, notably T cells and B cells and dendritic cells. These are immune cells that normally are in the circulation, but particularly in the gut and through the Peyer's patches, maintain an intimate contact with what would be the exterior of the body. So this is where a lot of the immune system education about what's going on outside of the body comes from. And this is where our immune cells learn about our exposures, learn about what we're eating, what we're smoking, what we're drinking, what we're exposed to. And there are several mechanisms by which the immune system and the gut can transmit that information to the brain. Even though they are anatomically distant, there are shortcuts that may facilitate this communication. Particularly, one of them is the vagus nerve. The vagus nerve innervates the intestines, and it goes straight to the central nervous system. So there might be some retrograde transportation of metabolites, small molecules, small proteins that can travel all the way up to the brain. And there has been experiments in which if you sever or transect the vagus nerve, then gene expression changes in the brain occur. And this is because of the absence of that communication between the gut and the brain. Another mechanism is, of course, the immune-related. So through immune responses, both adaptive and innate, the brain can know what's going on in the gut. Also, bacterial molecules and metabolites can be either transferred through the vagus nerve or through the circulation. So once they are in the blood, they can then pass the blood-brain barrier and get access to the brain, thereby stimulating or inhibiting certain pathways in the brain. So there is a long idea that the gut and the brain are connected, and this has been stimulated by the common belief of us having, for example, a gut sensation, or when one is nervous or anxious, that can be transmitted to gut and tummy aches and so on. So there are this intuitively lines of communications, but in recent years, this has become more and more known and less mysterious. So today, what I'm going to try to do is to show you some evidence that links the gut and the brain, particularly in the context of an autoimmune disease of the central nervous system, which is multiple sclerosis. So if one conducts a literature review of the last 20 years and looks for the words microbiome or microbiota, and I'll define them as microbiota is the community of organisms. If you've attended other lectures, you will know microbiota refers to the community of microorganisms that live in a symbiotic state with the host, with us. And the microbiome will represent the aggregation of genetic material from that community. So if one performs a literature review of the last 20 years with the words microbiome or microbiota in the title or the abstract of those articles, one could see an almost exponential growth starting around 2010. And you see, pay attention, this is a logarithmic axis on the Y. And so there's an exponential growth of articles referring to the microbiome or microbiota from the year 2000 on, but around 2010, diseases of the central nervous system started to be explored, and more. Every year, there will be doubling or even more of the number of articles that will involve the word microbiome or microbiota. And this involves not only multiple sclerosis, but also other neurological conditions and psychiatric conditions as well. So with this in mind, let me remind you that the human gut microbiota, which is one of the richest communities of microbes that live in and around us, is composed of about 10 to the 13 to 10 to the 14 microorganisms. This is more or less equivalent to 1:1, one bacterial cell per human cell. So we are about 50% human. Now, the collective microbiome, that means if we start counting the number of genes encoded by each of these different bacterial species, this outnumbers our own genome, our own number of genes by a factor of 100. So humans encode about 30 to 35,000 genes. The bacterial microbiome is composed of about 1 million different genes. So really, humans in this context can be considered superorganisms whose metabolisms represents really an interaction and an amalgamation of both microbial and human attributes. It is well known that humans cannot process certain foods, most notably vegetable fiber, and we need bacterial communities to do that, to extract those valuable metabolites that we need for our subsistence. That's only one of many examples of those metabolisms that are amalgamated. So really, if we think that we are our genes, then we're probably more our microbiome than our genome in terms of sheer numbers. So this is what really motivated me to go into this direction, coming from a background in genetics and realizing that the microbiome outnumbers our own genome by a factor of 100, I thought this cannot not be important for maintaining health and disease. So just a reminder of the tree of life, for those of you who are familiar with taxonomy. On the top here, if we are interested in the kingdom bacteria, then I'll be talking about phylum, and then the next is class, order, family, all the way down genus and species. I may be talking about genera and species more often, but this is just a reminder of the taxonomy. So just a reminder of how we came to know the importance of bacteria in health and disease. This goes back to the early 2000s when the National Institutes of Health launched the Human Microbiome Project, in which several volunteers provided samples from different body areas, body niches, and the bacterial content was sequenced through high throughput DNA sequencing technology. And those sequences were read and contrasted to existing databases, and we were able to identify which bacteria those sequences came from. And through that kind of analysis, we were able, for the first time, or the community of the scientific community was able for the first time to see how similar or dissimilar were different communities of bacteria coming from different individuals, but from the same niche. So if you look at all of this in the oral cavity, so these are about 100 different samples from 100 different individuals, and you can see that the samples cluster. So whenever two of these dots are closer to each other, that represents similarity across the entire DNA sequence of the microbiome. And you can see how the samples cluster by body niche rather than by individual. So all the gastrointestinal samples clustered together, all the oral samples clustered together, and all the skin samples clustered together, suggesting, again, very, very different bacterial niches in different body areas, which makes sense, thinking that different body areas have different pH, different humidity, different nutrients, and so on, that will allow the different types of bacteria to want to make a niche there. So one other aspect that I think it's really, really important is that this slide, also coming from the Human Microbiome Project, highlighted, is that if you look at the composition, and this is at the phylum level, so remember the tree of life, phylum is all the way almost to the top. So we're looking at big differences here. If you look at different individuals, and this each of these panels represents about 100 individuals, you see that the composition of the different phyla is wildly variable across different individuals within a particular niche. This is the nose, this is the mouth, this is stool, and so on. So you see high variability. However, when the same data is analyzed in the context, not only who's there, so which bacterial taxa is that DNA coming from, but what are the metabolic pathways that those bacterial taxa are capable of, then that becomes a lot more homogeneous. So you see now that across different individuals, now there's not such a big variation when one looks at the different biological and biochemical pathways. So what that teaches us is that, to a certain extent, it doesn't really matter who's there as long as they are performing a certain function. So we all need to process for example, vegetable fiber, but there are many different bacteria that can do that. So to a certain extent, it doesn't matter really which bacteria is doing the job as long as some is. So this is what this slide shows. And this is another interesting slide that relates to diet. And this is an interesting one because it really paints a clear picture of how much diet dominates the bacterial taxa that live in our guts. If we change our diets, the bacterial taxa that live in our guts will definitely change. This is a slide that was made by going to a zoo and collecting stool samples from different kind of animals and then sequencing their the DNA that was present in the stool. So identifying the bacteria in those samples. And then they're colored by the diet of the animals that those samples came from. And you can see how they clustered by whether there were herbivores or carnivores or omnivores. And this is a very, very interesting slide because, again, it shows the dominant aspect, the dominant role the diet plays in our bacterial diversity in the gut. So with this in mind, let's move on to now the interactions between gut bacteria, gut microbes, and the immune system. And in this regard, I think it's important to remember that microbes are mostly symbionts. And symbionts means they live in peace with the host. They're not pathogens. Most 99.9% of microbes are symbionts. They're not pathogens. So actually, we need them. We need them to have a healthy metabolism. And among the many functions that gut microbes play, they help, as I mentioned at the beginning, shape immune responses through the interaction of their metabolism with ours. And I'm highlighting a couple of interesting examples here. For example, the bacteria Bacteroides fragilis has been known to activate a particular kind of immune receptor called toll-like receptor 2 through a metabolite called polysaccharide A that is part of the bacterial wall. And that polysaccharide stimulates a special kind of immune cell called regulatory T cell that usually suppresses inflammation and enable its own colonization. So in a sense, it opens the door for the immune system to say, okay, I'm going to let you pass because I know that you're a good microbe, and then I'll suppress my immune response that otherwise I will have generated against you. Another example is a set of molecules called short-chain fatty acids, and these are metabolites that are produced by fermentation of dietary fiber. They regulate the size and the function of, again, this particular T cell population called Tregs or regulatory T cells, and can protect against many inflammatory events, among them colitis. So these are only two of many potential interactions between bacteria and the immune system under normal circumstances. Now, under normal circumstances, here depicted in the panel A, the microbiome, together with the genome of the host, which is unmodified throughout our lifetime, might determine the type of immune response that we will elicit. So in normal circumstances, when there is an equilibrium of in the gut microbiota, then there is an equilibrium in the type of immune responses, here depicted as the blue as the immunoregulatory Treg response, and the red as an inflammatory Th17 response. So when they're in equilibrium, then everything is in harmony. Now, however, our genome will not change, but the microbiome might change. So there might occur dysbiosis, whereby in this case, the yellow bacteria outnumber the green bacteria. And this is an oversimplified example, but in this case, if the yellow bacteria outnumbers the green bacteria, then the Th17 cell population, the red cells, are more stimulated because they respond more to the yellow bacteria, and then there's an imbalance of inflammatory responses towards a disease state. Similarly, the opposite could also happen, that the green bacteria, which are in this case the ones that stimulate the Tregs, might disappear in a given dysbiosis state, and in that case, then the Treg population suffers and diminishes, thereby again tilting the balance towards a pro-inflammatory state, which, if it's sustained over time, it may lead to disease. So this is an example of how dysbiosis in the gut, together with our own genome, together with our own innate ability to respond to pathogens or to certain kinds of bacteria, might determine our susceptibility to disease. So turning the page now and focusing more on the role of bacteria and the central nervous system, I would like to bring to your attention some studies that were conducted now a little over 10 years ago in the lab of Lloyd Casper, back then at Dartmouth University, where he showed for the first time that if you treat mice with wide spectrum antibiotics, then they become resistant to brain inflammation. And EAE here stands for experimental autoimmune encephalomyelitis. This is an experimental model of brain inflammation that resembles many aspects of human multiple sclerosis. So this discovery was really surprising, because for the first time it showed that you need a normal microbiota to produce an immune response against a brain antigen. So this was really groundbreaking at the time. And this effect was associated with a reduction in inflammatory cytokines and an increase in anti-inflammatory cytokines such as IL-10 and IL-13, and in the increase of the immunoregulatory population known as Tregs. So his lab also showed that oral administration of PSA, polysaccharide A, from these bacteria called Bacteroides fragilis, protected mice from EAE, and this protection was related to an interleukin-10 increased production. Another interesting fact was that if one monocolonizes a group of mice in which microbiota had been depleted via antibiotics or some other way, if one monocolonizes, so if one colonizes the gut of those mice with a single bacterial species, in this case, segmented filamentous bacteria. This is a mouse bacteria that usually colonizes mouse and not humans. Then investigators were able to restore the necessary Th17. Remember, Th17 is that pro-inflammatory response that is necessary to mount an immune response against a challenge that is given to induce EAE. So it only took segmented filamentous bacteria to restore that susceptibility after all bacteria had been depleted. So in this case, segmented filamentous bacteria was associated directly with stimulation of a pro-inflammatory immune response. So now we move on to more functional types of studies, and this was work performed by the laboratory of Hartmut Weckerle and colleagues in the Max Planck Institute in Munich. And what they've shown for the first time, this was also 10 years ago, was that they had engineered a mouse strain so that it will respond exclusively to a particular epitope, to a particular region of a protein known as myelin oligodendrocyte glycoprotein, MOG. And this protein is highly immunogenic. And MOG, it's only expressed in the brain. Therefore, if these mice are responding aggressively to MOG, eventually they will trigger an autoimmune response to MOG, and they will develop a disease similar to multiple sclerosis spontaneously, no stimulation of any kind. As long as you wait long enough, almost 80% of the mice will develop disease after 30 weeks of age. This is if they were kept under a specific pathogen-free conditions, SPF. What this is the regular mouse facility in a research setting, which is devoid of specific pathogens, viruses and mites and so on, but they, of course, allow the colonization of normal microbiota. So these mice have a normal microbiota in their guts, and if so, they develop the disease. However, if they are maintained in a germ-free state, meaning either by, again, keeping them on wide spectrum antibiotics, or like in this case, keeping them in a bubble, completely isolated from the exterior since birth, so that they cannot be colonized by anything. They're completely germ-free, completely sterile. These mice don't get sick. So again, showing that bacterial colonization of the gut is necessary to mount an immune response directed against the brain. And interestingly enough, when these mice were colonized, when these mice that will not get sick because they were germ-free, they were colonized with microbiota from their litter mates, they now acquire again the sensitivity to develop the disease. So this was a very elegant setting to show that the bacteria in the gut are necessary to mount an immune response against the brain. So now I would like to introduce the International MS Microbiome Study. This is a study that our group at UCSF is leading, and this is an international effort involving several centers in the US, Canada, Europe, and Argentina. And the goal of the International Microbiome Study is to become really a reference study in the field. And it has a very particular study design in which for each patient with multiple sclerosis, a household control is recruited into the study. And the reason for recruiting a household control is we want to become independent of all the other things that can possibly modify the gut microbiota, such as diet, most notably, but also, as you may have heard in previous talks, the presence of pets in the house, the presence of babies, whether you live in a rural setting or in an urban area, and so on. All of those may influence a composition of the gut microbiota independent of the disease. So if we recruit for every person with MS, we recruit a household control without the disease. If any differences in the microbiota exist, then we can associate them to the disease with higher certainty. So this is a very, very large study. We're aiming at 2,000 cases and 2,000 controls. And for each case, we collect stool samples, blood samples, and dietary questionnaires. Of course, we want to know what they're eating, and also a clinical questionnaire. We want to know how they're doing in their disease journey. So data that is coming out from that study, and this is, we're getting into immunology terrain, so I'll be slow here, are very, very interesting. And this was one of the first experiments that we performed. We wanted to know if microbiota from an MS patient is in any way different than microbiota coming from a healthy control with regards to the immune system. So does the immune system see microbiota from MS any differently than microbiota from a healthy individual? And the essay that we set up to answer this question was to culture together extracts of microbiota from patients or controls together with blood cells. And then we stimulated those blood cells, which contain lymphocytes, into a Treg polarizing condition. So we pushed those cells in vitro to become Treg. And we measure whether the presence of MS bacteria or healthy bacteria will affect in any way the development of these Treg cells, which are so important to control inflammation. And what we found was that microbiota from MS patients were less efficient at inducing Tregs than microbiota from healthy controls. So we didn't know whether they were, how different they were in terms of the composition, but functionally, the immune system saw them as very different. So the next step was, okay, now we want to know what is exactly the difference between MS microbiota and healthy microbiota. So, and this is a summary of a very large experiment involving several dozen individuals at this point. And each of these dots represents a bacterial taxa. And here we can see that on the right-hand side, these are the bacteria that are more represented among MS cases. And on the left-hand side, these are the bacteria that are less represented among MS cases. And here I'm highlighting a few taxa because we wanted to do functional studies with those to see what we could learn. And here we picked 3 genera, Acinetobacter, Akkermansia. These 2 were higher in MS than in controls, so more prevalent in MS than in controls. And then we chose Parabacteroides, which was significantly less prevalent. This is a log scale in MS than in controls. So then we perform similar assay than we perform at the beginning, trying to understand what happens if we culture or if we put together now a single species, now Akkermansia, in this case, Acinetobacter calcoaceticus, and we together with T cells, and then we differentiate those T cells towards Tregs, what would happen? And this is I don't know if you're familiar with this type of plot, but this is a flow cytometry analysis. And what this contour plot represents is the density of cells that's stained with 2 fluorescent markers. On the y-axis is FOXP3, which is a transcription factor typical of Tregs. And on the x-axis is the intensity of CD25, which is a membrane marker characteristic of CD4 positive T cells that are expressing the interleukin-2 receptor. So in this case, the Tregs are defined by those cells that are above the threshold of both CD25 and FOXP3. And in this case, if we don't put any bacteria, we can differentiate about 26% of those cells. However, if we incubate those cells with Acinetobacter calcoaceticus, that proportion gets significantly reduced. And this is a representation over more and more samples. So this is, if we look at many samples from many patients, then we can see that overall we see an inhibition of the capacity to stimulate Tregs when they are in contact with Acinetobacter calcoaceticus. Similarly, when we do the same experiment with another of the species, the bacterial species that were more represented among MS cases, we see that Akkermansia muciniphala increases Th1 lymphocytes. These are a type of lymphocyte that expresses large quantities of interferon gamma. Interferon gamma is a cytokine that stimulates an immune response. It's highly inflammatory. It attracts more cells to the site of inflammation. So we see that Akkermansia muciniphala significantly stimulates the production of Th1 lymphocytes. On the other hand, the bacteria that was less represented among MS cases, Parabacteroides distassonis, we found that when cultured with blood cells, it stimulates them to produce interleukin-10. So interleukin-10 is a cytokine associated with immunosuppression. So that makes sense. Again, the two bacteria that were found more prevalent among MS cases had a pro-inflammatory effect in vitro. The bacteria that was less inflammatory that was less prevalent among MS patients had an immunoregulatory effect, an anti-inflammatory effect. So the direction it made sense for us to start making a mental model of what might be happening. Of course, the question of, okay, is this cause or consequence always comes up? Are we really seeing these changes because of MS, or does MS happen because of these changes? And the best way that we have to answer this question is to conduct research with germ-free mice. And I've introduced that paradigm before. Germ-free mice are mice that are born in sterile conditions and kept in sterile conditions throughout their lives. And then we can colonize them with a bacterial sample of our choice. In this case, we colonize a group of mice, an entire group of mice with bacteria coming from a healthy individual, and another group of mice, we colonize them with bacteria coming from an MS patient. Then we induced EAE, and we observed whether the disease severity was any different between the two groups. To our surprise, we found that mice that had been colonized with the MS microbiota became more sicker than mice that were colonized with a control microbiota. And this, again, was a proof, the best we could do to prove the causality that these bacterial communities stimulate the immune system in a different way so that they have an effect on EAE, which is a model for multiple sclerosis. And we can extend that to our understanding of how multiple sclerosis might develop given a genetic predisposition to it. We might think that being colonized by a certain bacterial taxa, be that because of our lifestyle, our diets, our therapeutic profiles, the therapeutic drugs that we might be taking, all that may change the intestinal composition of bacteria so that they may stimulate the immune system in a different way. If we are genetically susceptible to that, that may lead to occurrence of disease. Another experiment that came out also from our colleagues that showed exactly the same thing. And in this case, these were mice. Remember, these were the engineered mice that will develop disease on their own. And when they colonized this, in this case, it was with twins. So these were discordant twins. They were genetically identical, but one had MS, the other didn't. And if they colonized a group of mice with the microbiota from the MS twin, mice became susceptible to the disease. If they colonized them with the microbiota coming from the healthy twin, the proportion of mice acquiring the disease was significantly lower. Again, different experimental setup, but very concordant results. So now I would like to close with our translational effort into trying to understand what might be a potential intervention that may modify the gut microbiota in humans so that to modify disease course. And in this case, this is a clinical trial, phase 1 clinical trial that we conducted using fecal microbiota transplantation in patients with relapsing multiple sclerosis. This was a very small trial, only 5 individuals that mostly in a phase 1, you're looking for tolerability and feasibility and make sure that you're not making more harm than good. But we also had a number of secondary and tertiary endpoints in which we're interested in knowing, for example, after transplantation, so these individuals were given a dose of antibiotics, they were given a bowel prep, and then through colonoscopy, they were administered microbiota from a healthy donor. And we were interested in knowing how long will the microbiota stay after transplantation. We were interested in knowing, does the recipient of the microbiota change in any way the immune response? Those are secondary endpoints of this trial, which we are right now analyzing. But the idea is that we're starting to move into to think about therapeutic and interventional ways in which we can modify the microbiota, particularly the gut microbiota, to see whether we can curb disease that either has already started, or if we can prevent disease, or we can prevent exacerbations of the disease. So with this, I would like to close and acknowledge all my colleagues from my group, all the colleagues from the UCSF Multiple Sclerosis Clinic, all our collaborators of the IMS-MS team, and our funding agencies. And of course, I'll be happy to take any questions that you may have.
Thank you so much, Sergio, for that really interesting talk. We have a couple of questions in the Q&A. So Sergio, we have just first a question for clarification. At the very beginning of your deck, you mentioned that there's kind of 100x more genetic material in the microbiome as opposed to our human genome. Is that across our entire human microbiome or just the gut microbiome?
Yeah, it's all right. I didn't make that clear. It's through the entire microbiome. However, the gut microbiome is the richest of all the niches. So the gut microbiome is where most of the bacterial diversity can be found.
Thanks, Sergio. The next question is about kind of sex differences. So can you comment a little bit on, what we know about the variation in both the composition and function of our microbiota across genders?
Yes, indeed. So yes, there are sex differences in the composition of the gut microbiota, and as the one might think, there are hormonal differences, and particularly after puberty, the sex differences are more marked. However, this is one of the reasons why we control statistically for this. So we know, so the statistical models that we use take the sex into account as a covariate, and then remove the effect from sex into the effect of the disease. So we're trying to remove, and the healthy control. Of course, most of our couples are heterosexuals, but we do have a few same-sex couples, which help us also validate that hypothesis.
Thank you. You might have covered this. Is there is multiple sclerosis more prevalent in, is there a sex prevalence?
Yes, indeed. Sclerosis? Yes. So like many autoimmune diseases, multiple sclerosis has a sex bias towards more frequent in women. Mm-hmm. In the case of multiple sclerosis, about 70% of the cases are women.
Great, thank you. How exactly is the vagus nerve interact with gut bacteria?
So the vagus nerve is the nerve that, again, it innervates the gut. So is where the electric impulses come directly from the brain into the gut for controlling peristalsis of the gut and help digestion. So because it is directly coming from the brain, there are bacterial metabolites that are very small molecules that are being produced, which can diffuse either passively or through active transportation by specialized proteins into the inside of the nerve, which is essentially a bundle of very elongated cells. And then once they are within the nerve, they can travel retrogradely all the way up to the brain, where they can exert their effect. So there's been really, really interesting studies, one that I can remember from Canada, where when they transected the vagus nerve in mice, changes in the expression of GABA receptors in the brain were observed, meaning that they're interrupting, by interrupting that flow of signal, whatever that might be, then the activation of genes in the brain can change. So that's one of very, very concrete example of how the vagus nerve can be a conduit for connecting the gut and the brain.
Thank you so much. So this question is asking generally about nutrition and the microbiome. So I know Peter Turnbach covered this kind of at length two lectures ago, but I wondered, can you comment what's known about, like, specifically if diet's been shown to improve or exacerbate MS flare-ups?
Right. Well, this is a topic of very, very intense research right now. I'd like to remind everyone that the field of microbiome is relatively new, and there's a lot to do. There's an incredible amount of interest in the population in general to see, okay, what diet should I do should I follow to have a healthy microbiome, to be disease-free, to be healthy, to be energetic. So we still don't know. There are very early times. These are very early times in the research, and more analysis of metabolic outputs are needed. There's, of course, dietary interventions are very difficult to enforce. So studies that involve a change in diet or maintenance of a particular diet are very difficult because, again, adherence to diets can be difficult to enforce and to follow. So metabolic studies, whereas the blood metabolism and the bacterial metabolism in the gut are measured, are the most direct experimental evidence that we can get to analyze very, very precisely what is the exact effect of diet on our metabolism. However, these studies are extremely expensive. So far they have been done in relatively small number of cases. As these studies become more affordable, I anticipate that there will be more and more studies shedding more light on exactly what is the role of diet, and particularly what is the role of specific nutrients, components of diet to curb or not disease. So far, most of the information that is out there, I must say, is largely anecdotic and not really evidence-based.
So we have a couple of questions asking if you could comment on kind of what's known right now about the link between microbiota and other neurological diseases. Parkinson's listed by one attendee, and dementia. So can you just spend a few minutes and kind of go over the literature for those?
Yes. So Parkinson's has long been suspected that the microbiota may have a role, particularly one of the lines of evidence is many Parkinson's patients complain of constipation early on in the disease, like several other diseases, but this in particular has been puzzling. And in fact, my colleague Sarkis Mesmanian at Caltech was one of the first to show the involvement of the gut microbiota in Parkinson's disease. He's done similar studies to the one that I described here, transferring microbiota from Parkinson's patients into mice that are germ-free. And he showed, for example, that mice receiving microbiota from Parkinson's patients have an altered cognition and altered behavior. So when they perform behavioral tests in these mice had altered behavior. So they went all the way to characterizing a particular protein from an organism that might be implicated in that. Still very early on, there's very active research. Similar in Alzheimer's disease and other types of dementia, similar in depression. It's really eye-opening to see how many correlations are now starting to be uncovered when more and more of these gut microbiome studies are being performed in neurological diseases. So it's very early times, but a very promising field indeed.
Mm-hmm. And are you aware of kind of any ongoing studies at UCSF similar to your multiple sclerosis house-dose studies that might start to get at those other kind of neurological diseases? Autism, Parkinson's is what the question specifically asks.
I see. I'm not aware of a similar microbiome study being done at UCSF for other neurological diseases at this time.
Yeah, not at this time. I know that there's discussions of those. As the audience might appreciate, it takes some time to roll those out. Turning back to multiple sclerosis, one question is, are there regional differences in the prevalence of multiple sclerosis? You talked early on about environmental determinants.
There are. However, these regional differences sometimes are difficult to pinpoint to what extent those are genetic versus really geographic differences. We know for sure that different ancestries, different genetic ancestries have different predisposition to multiple sclerosis. Multiple sclerosis is more prevalent in individuals coming from Northern European background, where the prevalence can be as high as 1 in 800 in Northern UK and the Scandinavia, and almost inexistent in Southeast Asia and Far East Asia. So that's mostly due to genetic ancestry. However, geographical location has also been implicated in epidemiological studies and attributing what is called a latitude effect. So this was very interesting studies done almost 30 years ago now, where individuals who migrated from a high prevalence area to a low prevalence area will carry the risk of origin if they migrated before puberty, after puberty. However, if they migrated before puberty, they will acquire the risk of the destination. Again, suggesting that the maturity of the immune system, wherever that takes place, even in a high prevalence area or in a low prevalence area, will determine your lifetime risk.
This question, I'm curious if you've gathered any information about stress levels in your experiment that has a member of the household of a person with MS as a control. I'm thinking there could be different levels of chronic stress between individuals in the household.
Yes. To a certain extent, this is something we indirectly can measure through metabolites, so cortisol levels and so on. However, this is a very tricky question because one might be stressed because of the disease and not have the disease because of stress. So again, this is one of those cause or consequence questions. An association may be found, but then to try to find out the causality, one will need to do additional experiments.
So Sergio, while we wait for a couple more questions, can you just comment on other kind of diseases that that your group is studying? And we haven't touched really at all on kind of integrating can you talk a little bit about spoke generally and the power that adding the microbiome data might be to that and our ability to prevent, predict, and treat disease?
Yeah, so again, I wanted to keep focus and not go very broad, but you're right. The work that we're doing on the computational side of the lab is very important because we really want to create a system that allows us to integrate the different data modalities that we gather from patients. Where I'm particularly interested in understanding the disease from an integral point of view, not just one aspect. And Spoke, in that sense, is a tool that allows us to integrate data. Spoke is a database of databases. It contains millions and millions of concepts related by their biologically meaningful links. So genes, diseases, we have 10,000 diseases, every gene in the genome, 5,000 bacterial organisms. All that information, all the metabolism, all the pathways is all in one place. So that allows us to, for example, embed data from a single patient onto Spoke, and then we can navigate through the microbiome, the individual's genome, individual lab tests that individual may have, clinical outcomes, symptoms, medications. So all that can be understood in the context of biology through something like Spoke. So this is relatively early, but it's a very, very promising technology that we're applying not only to multiple sclerosis, but to Parkinson's disease, dementias, and now we're getting into trying to use this to predict outcomes in many other chronic conditions.
Thank you so much. Two questions. Looks like final two questions have come in. One, is obesity a risk factor for development of multiple sclerosis?
Yes, this has been assessed by statistical and epidemiological analysis. There are several risk factors that have been identified in MS, and obesity is one of them. So this may have to do with again, cause or consequence with the dysbiosis of the gut and different immune responses between obese and lean individuals may have. So we know that in obesity, there's chronic inflammation, and over long periods of time, if you are genetically predisposed to an autoimmune disease, chronic inflammation will eventually end up in an autoimmune disease. So the short answer is yes, there is evidence.
So with that, we are going to go ahead and close out this lecture. Thank you for joining us. Thank you so much to Dr. Sergio Baronzini for his lecture, and we will see you next time. Thank you, everyone, and good evening.
Thank you. Good evening.